Hello and welcome back, Biology 182 lab students. We're going to be discussing Lab 2, the Kingdom Fungi, which is exercise 27, found on page 287 in your lab text. We'll be discussing various forms of fungus, including the common mushrooms, as we're used to, and of course, many mushrooms are quite famous, being represented in video games such as Super Mario Bros. Yeasts are also included in fungus. Molds, as we're used to them growing on bread or fruit that's been left out too long. Tooth fungus, which kind of looks like teeth in some cases. A coral fungus, which does look like coral, like stag coral. And then cup fungus, which usually have a cup shape to the reproductive structures. There's 56,000 species and counting, and they evolved around 500 million years ago. Your objectives to prepare for your test are, number one, what are the several ways fungus impact Earth's ecosystems? Number two, what traits are common to all fungi? 3, what traits are unique to each of the 4 phyla? 4, what events occur in the major stages of the alternation of generations in fungus? You should be familiar with alternation of generations from the previous lab. Is observed in algae, where we have a haploid and diploid stage alternating. 5, what are the specialized spore-producing structures and cells of each phylum? Most of us are familiar with some of the main ecological roles of fungus. Of course, we know that they play a major role in decomposition, and they're essential in breaking down dead organic matter. They use digestive enzymes secreted onto the substrate first, and then the nutrients are absorbed through their cell walls. It is opposite of us being a heterotroph, where we ingest the entire thing and then absorb nutrients within us. Their other main ecological role is nutrient cycling. So nutrients become trapped in dead matter like nitrogen compounds, certain carbon compounds. And most organisms cannot access these, but fungus can, and they make available to other living organisms. The nutrients return to the soil where plants can absorb them. So after the fungus is finished and it respires, then the plants can pick it up, they can eat it, and the nutrients pass to higher trophic levels. So basically, a lot of the nitrogen compounds that we need come from fungus. But here's a deeper question. What would be the repercussions in an ecosystem lacking fungus? And what are those main nutrients that plants depend on fungus to release back into the soil? Some additional ecological roles of fungus are mutualistic partnerships. I have a joke for you. What did the algae say to the fungus? I liken you. Lichens are composite organisms of fungus and algae, resilient, that can colonize harsh environments and begin succession by breaking down rock. We can see a picture of those off to the right. There's 4 different lichen species. Another interesting group are the mycorrhizae. Nearly all plants benefit from having this fungus on their roots, and they aid water and nutrient absorption. What do the fungi receive in return? So we can see off to the bottom right, mycorrhizae on plant roots. And then, of course, they are a food source for humans and ants. Leafcutter ants will go into the rainforest, they'll take pieces of leaf, and then they go into their colony and they begin to grow, like crops, very specific species of fungus which they eat. And some, they actually farm these fungus. Additional roles in an ecosystem that fungus plays are parasites. So they can be animal hosts like Chytridiomycosis, which we can see infecting that frog at the top right picture. It's a non-native chytrid fungi that comes from a poison dart frog that carries it naturally, and it is not a parasite onto that organism. But since those poison dart frogs have been a close association with other pets, say the American bullfrog, the bullfrog can also carry the chytrid fungus, and then people, when the bullfrog gets a little too big, they release it into the wild. And then the native frogs actually suffer massive die-offs. We can also see parasites in human pneumonia. Ringworm is not actually a worm at all. It's actually a fungus. An athlete's foot. And you can feel athlete's foot as it begins to grow its hyphae into our skin, touching our nerve endings. That's when your feet really burn and itch. And also plant pathogens. So a long time ago in Ireland, many Irish had to escape from Ireland because there was a great potato famine. And that was from a fungus that really caused a lot of starvation. And many of the Irish came to America, including my grandfather, and thus I am here because of him. They can also be predators. So they have sticky substances that can trap prey. And they can trap microinvertebrates with constricting rings. So off to the bottom left, we can see a nematode trapped by a fungal constriction ring. An interesting group is a parasitic fungus that has killed an ant after instructing it to climb to the top of a tree, called cordyceps fungus. The cordyceps actually take over the plant's or the ant's ganglion, which is much like its brain, and reprogram the ant to go to the top of a tree. And then the fungus sprouts from the ant's head with a reproductive body that produces spore that are carried by the wind. Another example of a disease is called the fungal Dutch elm disease, and it's spread by a beetle. And then another one that's a serious problem with chestnuts in America is the American chestnut blight that was caused by an Asian fungus. Of course, there are many economical uses of fungus. One is bioremediation. So many fungus degrade pesticides, herbicides, and radioactive elements like uranium and oil spills. So we can use these to clean up human messes. Isn't that kind of nice that nature can help us out from time to time? 2, food and beverages. We can ferment things with them. So our wine and beer, my lovely banana Hefeweizen, comes from a yeast. Oh, Hefeweizen. Cheese. I love cheese, and I don't really want to talk about it because I could go on for a while about cheddar and provolone cheese, but those are made by molds. Breads, again, are made by a yeast, which is the type of fungus. Drugs, so penicillin comes from the soil fungus Penicillium. And then LSD, which is an illegal drug but can be used for some types of research. That's synthesized from an ergot, which is a grain fungus. So we can see some pictures off to the right. We can use fungus to help clean up oil spills. We get wine from it, bread, penicillin, and cheese comes from molds. Oh. What is a fungus? Well, let's go over their traits. 1, they have eukaryotic cells. That means, of course, they have a membrane-bound nucleus and organelles, and they have a haploid-dominant life cycle. 2, they're absorptive heterotrophs. So absorptive meaning that they secrete the enzymes first and then absorb the nutrients the enzymes yield, and they do that in decomposition. They are spore-producing organisms. So the offspring are dispersed with either animals like the ant we mentioned before or wind. You'll have to forgive some of the noises. Mr. Pippins is preparing for bed, and he's making lots of scratching noises. And he makes a nest every night with his blanket. I can't explain that, but he does. It really doesn't look any better than when he first started, but he thinks it seems to be more comfortable, so I just go with it. 4, they're structurally supported with cell walls made of chitin. That chitin is essential to understand the difference between cellulose and chitin. We'll get into that a little later. 5, they're sessile. So they have a plant-like quality. They don't move around much. They rely on other means of transporting their spores around. They're built from filaments called hyphae. These are the fungal filaments. Collectively, we call them a mycelium, so when lots of hyphae are together. And the mycelium is a strong network of hyphae that can form an erect spore-producing structure, ones we're familiar with, mushrooms and puffballs. We can see off to the bottom left a mushroom that's quite brilliantly red color, a coral fungus that looks like a stag coral, and giant puffballs are found around here. But before you go picking mushrooms, please remember many mushrooms are highly deadly. So you need to consult with a mycologist and really know what you're eating before you put it in your mouth. Let's talk about spores and gametes. They're haploid, but they differ. So spores, N meaning haploid, are for dispersal of offspring, or clonal offspring. These are clones of the adult. They can be produced either asexually or sexually. One spore grows into an adult fungus. The gametes are also N, and these are required for sexual reproduction. Two gametes must unite to create a zygote with one combined 2N nucleus, one diploid nucleus. And then spores disperse offspring to new habitats, as we are aware. In the bottom left hand we can see a bread with the bread mold. We've got sporangia that produce a lot of spores. Then the spore germinates on a new bread, thus causing a new colony. And then gametes are used in sex. So these are a picture of human gametes here, but you get the idea. We have an egg and a sperm. Fungus are built from hyphae. I mentioned before that hyphae are these single cell in series, they're slender filaments of cytoplasm and nuclei, and they're enclosed in a cell wall. Sometimes they're separated by septa, and sometimes they're not. The mycelium is a massive branching. It's an interconnected hyphae that forms sex organs. Typically we don't see hyphae in the ground forming a mycelium because the idea of the hyphae is to absorb nutrients. So if they're bundled, they're not necessarily good at forming nutrients because they're surrounding each other. We can see the cell wall of one of these hyphae made out of chitin. See at the top right, we can see a spore, and then a spore growing a hypha. And then in the bottom left drawing of that diagram, we can see the mycelium. Or sometimes we have parasitic fungus that grow hypha with little intrusions of hypha called haustorium that go into living cells and take their nutrients. The mycelium themselves, as I mentioned, are groups of hyphae intertwined together, but you can see in the ground that root-like structure, which kind of are like roots, but not like plant roots. They're single in the ground to absorb nutrients. Let's compare asexual to sexual reproduction in fungi. Asexually produced spores are from mitosis in specialized structures called sporangia or conidia. These are clonal spores with genes from one parent. Sexually produced spores form after sex. So we'll have an egg and a sperm-like structure meet, and it'll make a zygote. Then this divides via meiosis to create genetically unique haploid spores. These are sexual spores, having genes from two parents. Something interesting about fungus is they can also reproduce via fragmentation. So if a hyphae is broken off of certain species of fungus, that piece can actually grow into its own fungus. Timing is important for sex. Sex occurs only when two genetically different hyphae contact, and environmental conditions are favorable. This is unlike algae. If you remember, they only did sexual reproduction in unfavorable conditions. Now that you're familiar with a phylogenetic tree or cladogram, let's look at one for fungi. Classification into phyla is based on unique sexual reproductive structures in fungus, the spore-producing bodies. So we find in fossils and rocks, again dating them with uranium dating, that they arose around 900 million years ago. They all had a common ancestor, probably something like a protist. And then this protist, ancient ancestor, produced spores. This gave rise to the fungus. So our most basic fungus are the Chytridiomycota. These guys are the simplest in form, and their reproductive structure seem to be a little simpler. The next group is Zygomycota, and these create an interesting zygote in between two of their reproductive structures. We'll take a look at those in a little while. And then Glomeromycota is something, another type of fungus. We will not be studying these in class, but they are a group you will need to know. It's paraphyletic, meaning we're not sure where it came from, if it came from a common ancestor or not. Ascomycota, these are the cup funguses. And then Basidiomycota, you guys are familiar with as mushrooms. Exploring the phylum Chytridiomycota, commonly known as the chytrids. Here's an interesting factoid. They're the most primitive fungus. They evolved in water 500 million years ago and are crucial decomposers of litter and dead organisms. How they get their energy is they're saprophytic, meaning that they draw their energy from dead or decaying matter and absorb with excreting enzymes, or they can be parasitic on algae or amphibians, as we discussed before. Habitat, we can find them in freshwater, moist soil, or on a host. Reproductive phases, they have sexual spores that are released into the water from a chytridium, while asexual spores are released from sporangia. They have primitive traits, so 1, they're aquatic, or semi-aquatic. 2, they're unicellular and filamentous without any mycelium. 3, they have flagellated spores and gametes. And 4, they have a simple life cycle with haploid stage. So we see off to the right a unicellular chytrid, and then the bottom right is Allomyces, a multicellular chytrid with sporangia. Looking at Phylum Chytridiomycota, the unique traits that you'll need to remember for this phylum are: 1, they have flagellate spores. They're vital for dispersing offspring through aquatic habitat. They also have flagellated gametes, so that's a sperm and an egg that find each other for external fertilization. They have what's called a chytridium. It's a sexual spore-producing structure only seen in this phylum. And then we have no mycelium that's formed, but rhizoid hyphae anchor the fungus to its substrate. In the right-hand picture we can see a picture of the chytridium, and we can see those rhizoids, those anchoring hyphae that keep them all together. In the alternation of generations, there are 3 life stages you'll need to know. 1 you're familiar with, haploid. Haploid stage is much like our gametes, but in fungus it's the dominant stage in their life cycle. Each cell has 1 set of chromosomes and 1 nucleus. So that means each cell only has 1 allele per gene. The dikaryotic stage is an interesting stage where 2 hyphae from different organisms will meet, 2 different organisms from the same species will meet, and their cells will fuse. And then we'll have this kind of larger cell with 2 nuclei in it. But the nuclei do not fuse. This is the sexual stage in most fungi that forms a spore-producing structure. So this is the mushroom phase. It occurs when conditions are moist and humid, favoring spore dispersal. Each cell has 2 separate nuclei, meaning that each nuclei is different genetically. They have 2 completely different set of alleles. And both of those came from each parent fungus. We call these strains A and B. The diploid stage you're familiar with. It's an ephemeral stage occurring in specialized cells, for example Basidia, that immediately precedes spore production via meiosis. So the 2 nuclei in the dikaryotic stage will fuse. Then we have a 2N group, and then that 2N will go through meiosis, producing 4 gametes, and then each cell has 1 nucleus with 2 sets of chromosomes from each parent fungus. So what we've done here in that diploid stage is we've successfully mixed our genes, kind of like shuffling cards, so that no 2 players get the same cards in 2 different hands. Looking at the Phylum Zygomycota, these are the bread molds. We're familiar with these on our bread. Their habitat is soil, fruit, organic matter, and some animals like frogs. Their energy, again, they can be saprophytic, which means they draw their nutrients from dead or decayed organic matter, and they can be parasitic. The reproduction, so they have asexual reproductive structure called the sporangium, and these release clonal spores, while the zygosporangium, as a special type of sporangium, is the product of sex and produces and releases sexual spores, offspring, typically into the wind, although some of these actually explode and fire their spores at very incredible rates. They have a unique trait, that zygosporangium. It's a dormant stage that's produced in times of stress. So this structure is dikaryotic, and that's where fertilization occurs. When conditions are ideal, it releases the sexual spores via an emergent sporangium. Off to the right, we can see a fruit with 2 different colonies of bread molds that have grown on it. Then we can see our typical bread, and it has the sporangium, the sporangiophore, the stolon, the spores, and the rhizoid. And then in the bottom right-hand picture, we can see that zygosporangium, very specific, only grown in times of stress, and it's dikaryotic, so it has 2 nuclei that have not fused. Now let's look at the Zygomycota sex and the alternation of generations with 3 stages. Let's examine our right-hand diagram first before we get into real terms. I want you to notice the haploid stage is in pink, the dikaryotic stage is in yellow, and the diploid stage is in blue. Alright, number 1, the fungal strains A and B, so from 2 different individuals in the same species, unite. Each produce gametes, or gametangia. 2, fungi A and B unite their cell walls, something called plasmogamy, but not their nuclei, creating a dikaryotic mass called the zygosporangium. When conditions are good, fertilization and meiosis occur inside the dikaryotic cell, and then this makes a diploid zygosporangium, and then meiosis occurs and a sporangium emerges out of the zygosporangium and releases the offspring. And we call these the sexual spores. The reason why is those spores were directly produced via sex, not cloning. This is how zygomycotans actually shuffle their genes. So we shuffle our genes all the time, but a quicker way of reproducing is just via mitosis. Then you don't have to find a mate. And that works for only so long. Then you're relying on mutation only for introduction of new genes. But if reliant on mutation only, you can imagine a population won't evolve very quickly. So, introducing sex into an asexual lifestyle allows for crossing over and independent assortment to occur, thereby shuffling up the genes and really changing the genome of an organism so that it can evolve quicker. Now we're going to go on to the phylum Ascomycota, which are the sac fungus or cup fungus. They're the largest, most diverse group of fungus, ranging from unicellular and yeasts to large morels. We can see that in the top right-hand picture, and then just below that is a yeast picture. Their habitat, they're mostly marine, freshwater, land, and on a host. Reproduction, their spores are released from asexual conidia and something called a sexual ascocarp structure. Their ecology is many species exist in mutualisms with other organisms, such as lichens. Some species live within plant leaves and defend the plant from harmful pathogens. Their unique traits, they have an ascocarp, the asci, and the ascospores. We can see several different examples. Our cup fungus, our truffles, many of us enjoy those covered in chocolate. Those are the actual ascocarps. And then yeast produces something like that. And there's another cup fungus off to the bottom left. Let's take a look at the asexual reproduction in conidia for the Ascomycetes. The conidia are clonal spores that form in chains via mitosis. and are released from elevated stalks that arise from the mycelium. So in the left-hand picture we can see the conidia in a scanning electron micrograph. We can see the conidia 4, and then in the top, or in the right-hand picture, we can see the conidia and the conidia 4 in a light microscope that has been stained to show the contrast between the conidia and conidia 4. The sexual reproduction of ascomycetes is their dikaryotic stage, so they have 2 nuclei in the ascocarp. The ascocarp is the spore cup, so it's a sexual spore-producing cup that produces spore sacs called asci. The ascus, and the plural of that is asci, is the sac where spores develop. There are 8 spores per ascus, and those spores are called ascospores. That is something very unique to this group, hence where it gets its name Ascomycota. Off to the right we can see some asci with those sacs with 8 spores each. So we'll have meiosis go on once, and then mitosis happen once, and end up with 8 spores. We can see in the left-hand picture the asci line the ascocarp's rim, and spores will emerge. So we have developing meiotic spores, and then the mitotic group goes through mitosis, and we see a mature asci, and then sometimes we'll see sterile hyphae that don't have any asci in them. Why they're cup-shaped is typically to be dispersed by rain. Rain can go in one side, a raindrop, and then blast out all of the pollen on the other side. The alternation of generations have 3 stages again with the ascomycetes. Let's look at our right-hand diagram and just look at the basic structure. So the haploid is pink, the dikaryotic is yellow, and the diploid is blue. So what happens is 2 fungus meet in the soil, and the hyphae cell walls unite but not the nuclei, again plasmogamy, yielding a dikaryotic mycelium that forms the spore cup or ascocarp. The sacs or asci form around the edge of the cup. Then 5, the karyogamy, so we actually have the fusion of the nuclei and each ascus, so a brief diploid stage. Then meiosis yields ascospores, so remember 1 per cell, per zygote, 1 phase of meiosis and 1 phase of mitosis, yielding how many ascospores per asci? 8. So ascospores then are N and are dispersed via wind blowing over the top of the spore to the spore cup. Probably the most recognized phylum of fungus are the Basidiomycetes, and these are the club fungus. We find these guys and how they get their energy. They can be parasites, and then they can be terrestrial saprophytes, which means, again, we can find them taking dead and decaying organic matter and recycling it. Many mushrooms emerge from underground, so you'll see them actually sprout through the ground. The unique traits and reproduction of them. So they have a basidiocarp that is called a mushroom cap. That's a sexual reproductive structure, kind of like our ascocarp from the previous phyla. They have gills on the bottom that hold spore cells called basidia, which produce spores or basidiospores. The ecology of them, this includes rusts and smuts, which can be a big problem on many plants, some of which have caused a lot of tree mortality, so like white pine blister rust. In the bottom left-hand picture, we can find a puffball that grows around here in Flagstaff, and they can be very large, and when stepped on, the spores come out in a cloud, hence the name puffball. On the underside of the mushrooms, we can see those gills. In the top right, we can see another type of mushroom called earth stars. And shelf fungus many times grow on a dead tree trunk that's still standing erect. In the bottom right-hand picture, we can see the basidium, and those hold 4 brown spores. The sexual reproductive structures in Basidiomycota are the basidiocarp parts. So we have the pileus, which is called the cap. It's at the top. The gills underneath, those are tissue layers bearing spore cells. The basidia themselves, those are the spore cells. And this is where sex occurs. So fertilization changes the basidium from dikaryotic to diploid. And then that's followed by meiosis, which yields haploid spores called basidiospores. And these are sexual spores produced from meiosis in the basidia. In our top right-hand diagram, we can see our pileus. We can see the ring. We can see the gills underneath, or something sometimes called lamellae. The scales are on top. We have something called a stem, and then the cup that holds the stem, and then we have the mycelial threads and then hyphae that spread out from that. We can see the basidia, and since we only go through a phase of meiosis, and we don't go through that second phase as seen in Ascomycota of mitosis, we yield 4 basidiospores that are all haploid at this point. As I mentioned before, the sex occurs in the basidia cells. So this is where karyogamy happens. There's a video in the top right hand. So let's take a look at our diagram again. Let's split it up into our dikaryotic phase, our diploid phase, and our haploid phase. Starting with the dikaryotic phase, we can see the development of our basidiocarp. As it grows, it gets its gills lined with basidia. That's where karyogamy occurs. Kind of like fertilization. So the nuclei unite to form a diploid zygote. It doesn't last very long, so we have this zygote inside there that goes through meiosis, leaving the diploid stage behind and going into the haploid stage. Then we have 4 basidiospores, as you can see in the bottom right-hand SEM picture, and they hang onto the bottom of those structures, ready to be carried away by the wind. So then the wind will carry the spores, they'll plant in the ground, and then they'll each grow their own hyphae. Now, fungus A and fungus B, those hyphae meet, and then we have, again, the same system. Plasmogamy occurs first, and then that's a dikaryotic structure that forms the basidiocarp, and the cycle repeats itself. The sexual reproduction in Basidiomycota, so one, we see the parental hyphae unite, and their dikaryotic mycelium emerges from below as a basidiocarp. These are the familiar mushrooms. And of course the dikaryotic club-shaped basidia form gills. And then within each basidium, a diploid zygote forms and then immediately undergoes meiosis, yielding 4 basidiospores. And then those basidiospores are spread to new environments via wind and sometimes via water. Spores land and germinate into hyphae. So off to the right we can see a basidiocarp section with yellow gills. What is a lichen? Well, as I mentioned before, they're a composite organism of alga and fungus. The relationship in lichens is thought to be mutualistic. We know that the fungus receives sugars, so photosynthate from the algae. However, we have yet to observe that the algae are receiving nutrients, minerals from the fungus. Nonetheless, the algae are provided a habitat outside their otherwise aquatic or semi-aquatic environments. Lichens are very sensitive bioindicators for pollution. So we look around and we find these guys on rocks, especially around Flagstaff, and we can tell what particulates are in the air. The symbiotic relationship between green algae or cyanobacteria, so it can be both green algae, cyanobacteria, or singularly each one. And then the fungus, usually an Ascomycota fungus is ones that form these, although others can do it as well. They absorb minerals through the air and their substrates, especially rocks. They go through sexual reproduction. The ascoscarps are produced in the lichen body and appear as small cups which release spores. To the left we can see some bark lichens that grow on the bark of trees. Sometimes we find them on the Arizona sycamore. Then we can see to the right hand picture one growing on a branch. It's quite foliose. And then in the bottom picture we can see a crustose that's lichen that's very close to the rock. And there's a joke. Annie Alga met Freddie Fungi and they took a liking to each other. I should write a song about that, huh? I think I will. Crustose lichens can't be removed. They're really close to rock. These lichens have adhered so tightly to their substrate they cannot be removed from it. If you look closely, you'll see round spore cups that are mini ascocarps called apothecia. You can see to the bottom left picture a tree bark lichen growing very close to the tree bark, and then we have rock lichens. They come in many colors. Around here in Flagstaff we can see yellow, red, green, white, tan, blue, gray. Foliose lichens are leafy. So these lichens are like a leaf having two surfaces, a top and a bottom. They can be removed from their substrate. Many species have flat growth form but have frilly edges that lift off their substrate. Tree lungwort is an important bioindicator of pristine forest habitat, and it's very sensitive to air pollution. So that you can see a picture of that tree lungwort in the left-hand picture. And then we can see a little bit more of foliose lichen growing over moss. Fruticose lichens stand tall. These lichens project off of their substrates. They have erect reproductive stalks or thread-like bodies that often drape off trees or rocks. So an example of this are the British soldier lichen with red spore cups, and we can see that those red spore cups are projecting quite far from the ground. We can see down in the bottom right, pixie cups. Those release spores. And then Spanish moss isn't a moss at all, seen in the southeastern United States. It's actually a lichen. In summary, fungus are a diverse and ubiquitous group of simple organisms that play a major role in nutrient cycling. Most of their lives are spent underground or within their substrates, but when it's time to reproduce, they can form macroscopic spore-producing structures, the key trait in their classification. Fungus use clonal and sexual spores to transport clones and their offspring, respectively, to new habitats via the wind or sometimes water. Although sessile, fungus are more closely related to animals, feeding by decomposing and then absorbing nutrients from their substrate. They have complex life cycles that include haploid, diploid, and dikaryotic stages, and the latter of which is unique to the kingdom fungi. So in the right-hand pictures, in the top right, we can see basidiocarp gills. We can see a bunch of basidiocarps as they're growing on a log and decomposing the cellulose. And then Amylaria is an 8,000-year-old clone.
Close your eyes, I can find a new way to hope for you. You're in demise, I tried to find a new way to bring that light back to your eyes. I know you, I can see you.